So girls, in the last class we had discussed on this Ectocarpus, which belongs to the members of the family which are characterized by simple heterotrichous thallus, remember, which are referred to as the brown algae. And we had already discussed on the thallus structure, the cell structure, as well as the growth. So today we will be discuss on reproduction of Ectocarpus. Now this species Ectocarpus, it reproduces by asexual and sexual methods. In asexual reproduction, it usually takes place by a biflagellate zoospore. Here you can see in this figure, okay, you can see here in figure E, you can see this is a biflagellate zoospore. Okay, so these Ectocarpus, these species, these algae, they usually reproduce asexually by means of these biflagellate zoospores. Now, the sporophytic diploid plant, it produces two types of sporangia. That is, this what also I had mentioned in the last class, if you remember. The first type of sporangia is referred to as the unilocular. You can see here the unilocular sporangia, and the second type is referred to as the plurilocular or neutral sporangia. Unilocular, single chambered, and plurilocular, as you can see here, pluri meaning many. Okay, so both these types of sporangia, as you can see in this figure here, this plurilocular as well as the unilocular sporangia, they are born on the same filament, on the same plant. Okay, but there are some species where these two types of sporangia, they are formed on different plants, on different plant body. Now the unilocular sporangia, they usually form haploid zoospores. Okay, they produce haploid zoospores, and whereas the plurilocular, they will develop diploid zoospores. So now let us study about these sporangia one by one. Now coming to the unilocular sporangia first, these sporangia, they develop singly. Let me show you this figure. You can see here in figure A, these sporangia, they develop singly at the tip of the small branchlet. Here you can see in this figure A, at the tip of the small branches, small branchlets, the terminal cell here you can see at of the branchlet, it enlarges in size. Okay, it will enlarges in size and it will become the sporangial initial. Alright, so you can see here, this is the sporangial initial which will form on the which will develop at the tip of the small branchlet, and this will enlarge in size and act as a sporangial initial. Now the first division of the diploid nucleus is meiotic. Okay, so the nucleus here, it will divide meiotically, and then it is followed by mitotic division. So this will result in the formation of 32 or 64 haploid nuclei. Okay, so you can see here it is dividing, this sporangial initial, it is dividing into two cells. Here we have the sporangial initial. When it further divided, you can see here this small cell here below, it is referred to as the stalk cell. Okay, whereas the nucleus, it will first divide by meiotic division, and then it will followed by a mitotic division, forming about 32 or 64 haploid nuclei. You can see here many nuclei inside. This sporangium has been form. Now when the nucleus is divided, okay, it is not accompanied, it is not followed by the wall formation. There is no wall formation. As you can see, these nuclei, they are free here inside, they are present freely, there is no wall between them. So just remember, there is no wall formation which is accompanied with nuclear division, and as such the sporangium, it remains unilocular. That is why here it remains unilocular, meaning single chambered, okay, because there is no wall formation between these nucleus. But the protoplast of the sporangium, it will start dividing, okay, it will divide into as many segments as the number of nuclei. Suppose here there are about just an example, there are 50 nuclei, then the protoplast, it will form 50 segments, okay, in order to separate these nuclei. And each segment, it metamorphoses, or it develop into a pyriform uninucleate zoospore. You can see here pyriform meaning pear shaped, okay, pear shaped uninucleate zoospores. These zoospores, they are uninucleate, meaning having single nuclei. And the zoospore, it has a chromatophore and an eye spot. Okay, the chromatophore, that is the pigment containing cells. You can see here along with these nuclei, there are many chromatophores, and thus when the protoplast is divided into many segments, okay, and finally each segment, it metamorphoses into a pyriform or a pear-shaped uninucleate zoospore. So each zoospore here, it has a chromatophore and an eye spot. An eye spot, I had already mentioned before also, that it is a photoreceptive cell which sends the light direction as well as intensity, okay, the intensity of light as well as the direction. Now you can see here the zoospore, the two flagella, they are here in figure E. The it is biflagellate, all right, it has two flagella, but these flagella, they are laterally inserted and they are unequal, okay. You can see here the anterior one, this anterior flagella is longer and pantonymatic. You can see here there are many small fine hairs on this flagella, so it is pantonymatic. Whereas the posterior one, it is shorter. You can see here the one below, it is shorter and it is smooth, okay, there are no fine hairs on this one. On this shorter one, it is smooth and it is acronematic. So one is pantonymatic and the other one is acronematic. So these zoospores, they are then discharged between 600 and 1200 hours, okay, they will discharge, they ooze out of the sporangium in a gelatinous matrix. So here they will be released from this sporangium, okay, sorry, these zoospores, then they will be released from the sporangium through a small terminal perforation. There is a perforation which will form on this sporangium in order to release the zoospores outside, and they ooze out of the sporangium in a gelatinous matrix. Now after they discharge, they are not directly exposed into the environment. Instead, they remain in a spherical mass, okay, just at the apex of the sporangium there is a spherical mass which will form, and these zoospores, they will remain in a spherical mass at the apex of the sporangium for some time, for a while, and then they will become free. After that, they will become free, they will be released and swim off individually. And these, when they are released into the outside environment, they will swim off individually, and they seldom remain motile for more than 30 minutes. Okay, so you can see in figure F that these each of these zoospores, they withdraw their flagella. So here they are deflagellated. What they don't have flagella, they already withdraw their flagella, and they remain motile for more than 30 minutes. Okay, after they swim, then they will remain motile, they will withdraw their flagella, and they will attach themselves to the substratum by the anterior ends. Okay, so they will attach to the substratum by their anterior ends and start germinating within 2 to 3 hours, and when they germinate, they will finally give rise to haploid filaments. And sometimes after the liberation of the zoospores, a new sporangium. Okay, so these just remember, these zoospores, after they attach to the substratum, they remain motile, they attach to the substratum, they will start to germinate within 2 to 3 hours, and they will produce haploid filaments. But what happened to this one after some time? Okay, sometimes after the liberation of these zoospores, a new sporangium will initiate. Okay, initiates within the same sporangial wall. So within the same sporangial wall, a new sporangium will form. So this process will continue. So this is the unilocular, how these unilocular sporangia are formed. So next is plurilocular, or also known as neutral sporangia. Now just like the unilocular sporangia, you can see here in figure A, these sporangia, they also develop from terminal cell of the branchlets here. Okay, they develop from the terminal cell of the branchlets of diploid thalli, and the terminal cell, it acts as a sporangial initial. Alright, it will act as a sporangial initial and enlarges and becomes spherical, and it accumulates numerous chromatophores. So just like the unilocular sporangia, you can see in this figure, it accumulate numerous chromatophore. In figure C here you can see. So even this, similar to the unilocular sporangia, it accumulates numerous chromatophore. And now the sporangial initial, it undergoes repeated transverse and vertical division. So unlike unilocular sporangia, we see here that the nuclei, they develop, the nucleus, the diploid nucleus, it divided meiotically and mitotically, but the nuclear division, they are not accompanied by wall formation. So that is why it is unilocular. It remains in a single chamber here. Alright, but the difference in plurilocular sporangia is that the sporangial initial, as you can see here in this figure B as well as figure C, the sporangia, it undergoes repeated transverse as well as vertical divisions, forming as many as 660 cubical cells. You can see these are the cubical cells which are formed due to the division of the sporangial initial. And these cubical cells, you can see here, they are arranged here in mature plurilocular sporangium. You can see they are arranged in definite tiers, okay, in definite rows. And this multicellular structure is known as plurilocular sporangium. Now each diploid uninucleate cell, okay, this single cell here, it is uninucleate. You can see in one cell only one nucleus is present. That is why uninucleate. So this uninucleate cell, it will, of the sporangium, it produces a single biflagellate zoospore. So you can see, just like in the case of unilocular sporangia, here also biflagellate zoospores are produced. But the only difference between the zoospores produced by unilocular and plurilocular sporangia is that in unilocular, we have already studied that they they are haploid. The zoospores are haploid. They will form the haploid filaments, whereas here in plurilocular sporangia they are diploid. So diploid zoospores are formed in plurilocular sporangium. So just remember that is the difference. Now the mature zoospores, just like in the case of unilocular, they are liberated from the sporangium again by a terminal or a lateral by a terminal or a lateral pore. So you can see a pore will form and they are liberated. Now when they are liberated, they remain motile for 3 to 5 hours. Okay, and then they will germinate to form diploid asexual thalli. So in case of unilocular, we get haploid filaments, but in this case the zoospores, they will germinate to form diploid asexual thalli, and at maturity these thalli they bear both unilocular and plurilocular sporangia. So when they mature, when they develop, as you can see here in this figure B and C, in both these figure, these these diploid thalli, uh, they will bear both unilocular as well as plurilocular sporangia. You can see both are present. Okay, now since these diploid zoospores, they help only in the multiplication of the sporophyte, they do not play any part in alteration of generation. So when we discuss later the alteration of generation, you will see that these diploid zoospores, they do not play any part in alteration of generation, since they only help in the multiplication, okay, of the sporophyte. Now the formation of the plurilocular and as well as the unilocular sporangia by a species, by this Ectocarpus species, it is affected by certain parameters, okay, such as temperature as well as the salinity of water. For example, uh, I'll give you one example of Ectocarpus, that is Ectocarpus siliculosus. This particular species, it produces unilocular sporangia at 13 degrees centigrade. So when the temperature is 13 degrees centigrade, it form unilocular sporangia. But what happens when the temperature it rises to 19 degrees centigrade? It will produce plurilocular sporangia. But when the temperature is at 16 degrees centigrade, then both unilocular and plurilocular sporangia are formed. So you can see that we can see that it is affected by the formation of these types of asexual spores, are asexual structures, they are affected by temperature. So now we will take a look at the next topic, that is the sexual reproduction.